In this video, we will discuss about how the irregularities in intracellular chaperones might play important role in hemostasis. This hemostasis means stop in the blood flow circulation because of many reasons. So this study focuses on the importance of intracellular chaperones on hemostasis. So introduction starts with the why protein hemostasis is important. Protein hemostasis is important for maintaining the equilibrium appropriate amount of proteins within the cells for cellular processing, and any imbalance will lead to disease. One of such examples shown is the Parkinson disease. So what happens in the Parkinson disease? The dopamine amount of dopamines get reduced. Because of this, the signal transports between or message transport between two neurons gets affected, and one of the possibility is that the dopamine associated, or we can say dopamine transporter, it is a glycoproteins that are usually present on the membrane of the neuron, and with ages, if with ages it has been this Parkinson disease are been usually found in the aged person. Uh, so it has been found that with age, cellular functioning gets affected, and cell is not able to perform its function properly. And in one study, it has been found that the glycosylation of the this dopamine transporter will affect the transport of message from one neurons to another neuron, because dopamine will not interact with dopamine transporter. So this is the motivation that glycosylation might play an important role in, or glycosylation, or we can say in a more broader view, the post-translation modification plays an important role for normal functioning of the cell, and any abnormalities in this post-translation modification will lead to diseases, and one of such diseases is hemostasis. So in order to understand these things in details, the study focuses on finding out the global impact of intracellular glycoproteins such as this Calnexin and Calreticulum in the Calnexin and Calreticulum cycles that we have discussed in previous videos, that it is the initial point for asparagine glycosylation in endoplasmic reticulum. So this study focuses on used four kind of methods. First one is the proteomics method. From the proteomics method, we have identified, or we did the relative quantification through this iron star method that has been developed by QLab. And from this proteomics, we have found out that here. From this proteomics, we have found out that in Calreticulum knockout, the pathways that are related to the ER stress got down regulated in Calreticulum knockout compared to the other knockouts. So with this, we get a preliminary idea that knockout of intracellular glycoprotein chaperones in endoplasmic reticulums will cause ER stress, and that too at different level. So in order to confirm this, we did the ER stress assay, more precise ER stress assay. So in this, when we did the more precise ER stress assay, we have found that Calreticulum has the highest ER stress compared to other knockouts, and this thing is further confirmed with the expression of CHOP protein. CHOP protein's size is 27 kilodalton. We can see that in Calreticulum knockouts, the ER stress is maximum compared to the other knockouts. So from this, we can conclude that ER stress is higher in Calreticulum knockouts compared to other knockouts. Then we go further to verify whether this is related to any kind of disease or not. So we did the cell surface glycoprotein analysis, and then we have find out that CD44 proteins, or we can say the cell surface glycans, in case of Calreticulum knockouts, contains more signature for CD44 proteins compared to the other knockouts. So this CD44 proteins is also a signature for the cancerous cells. So from this it seems like if calreticulum is not functioning properly, it might switch itself towards more switch itself and behave more like a cancerous cells. So and these things might even align with the previous finding that with age the cells might deteriorate and people might get disease, and one of such disease could be cancer. So from this it looks like calreticulum improper functioning of calreticulum might promote or might help the cancerous cells. And next thing that we did, we also analyzed the cell cycle of the each knockout with the help of propinium iodide. And what this cell cycles analysis shows that in the wild type, the first portion, that is P3 P3 portion represents the G0 or G1 phase, and P4 portion represents S phase, and then P5 portion represents the G2 or M phase. So in this we have find out that in Calnexin and in case of double knockout, the cell cycle seems to be arrest in between G2 M phase. So, and these kind of conditions are also similar to the condition that has been found in this chronic myelogenous leukemia cells. In these cells also we have find out, the literature has described that there is a condition in which the proteins, that is DNA damage inducible protein GADD45 alpha, is overexpressed, and in case of cell cycle arrest at G2 and M, the proteins that is responsible for this condition, that is cell cycle arrest as G2 and M phase, is the same protein that is DNA damage inducible protein alpha, or GADD45 alpha. And these things the tabulated form of this graph is shown on the third panel. In this we can see that in case of double knockout, the M phase is increased to 45% compared to the wild type, that is 33%. In case of Calnexin knockout also it is increased to 42%. So this shows that the cells that are double knockout and the Calnexin knockout will behave more like this chronic myelogenous leukemia cells. So both of these finding indicates that this indicates that the Calnexin and Calreticulum both plays an important role in maintaining the normal functioning of the cells, and any disruption in this intracellular glycoproteins might lead to diseases, and that could be happening in the aged person, as with time the functioning of the cells deteriorate, and one of the things that might deteriorate is the glycoproteins, or we can say glycosylation formation, as this thing is summarized in this graph, that is in this figure, that is each person with time the cells functioning can deteriorate, and this cell functioning could be related to improper glycosylation, or more specifically improper and linked glycosylation, that is calreticulum or calnexin might not be functioning properly, and this might lead to diseases that aged person has observed. And the conclusion is that the more information that we the more system level study or knowledge or information we can get about how this glycosylation pattern changes in case in the diseased persons. That information might be helpful in reversing those disease condition, or you can say to cure those diseases in the old age person. The final figure shows the fluorescence image. The fluorescence image of different knockout cell lines when they are uh, transfected, or we can say lentivirus, when the stable transfection of different knockout cell lines are being done using reporter that is PLVX ATF4 PLVX ATF4, that can represent or that can display the real-time stress within the knockouts. And we can see that in Calreticulum knockouts the signals are quite high and is comparable to the positive control, that is wild type, when it is wild type in which ER stress is induced with the help of thapsigargin, that usually disturbs the calcium homeostasis. So the whole point of this study is that glycosylation is one of the important biological activity, and disruption in this glycosylation might lead to several diseases. So because of this, it is essential to understand the basic functioning or system level study of this glycosylation, and how they are, and how individual factors or components within this glycosylation uh, process plays a role on the maintaining the overall cell functioning of the cells, so that so that this information might help help to cure the persons who are infected with such diseases. So that's all for this video. Thank you for your time.